In this video, we will discuss about some of the approaches, the therapeutic approaches to prevent SARS-CoV-2 to infect human beings. Before going to the main topic, let us just quickly revise how SARS-CoV-2 replication cycle works. So, it begins with SARS-CoV-2 virus uh, interact with the receptor present in the plasma membrane of the host, and this receptor is usually, in case of human beings, is ACE2 receptor. So when it binds with ACE2 receptor, there is a proteases proteases formed by the host cell that helps in the invasion of the SARS-CoV-2 virus inside the cells, or in scientific term we can call it like endocytosis occur uh, at this point. After that, uh, the viral genome, that is positive single strands RNA that is present within the SARS-CoV-2 virus, is released and is been translated by ribosome. So here translation of viral polymerase protein occur, and this viral polymerase protein contain not only the structural proteins, but also the non-structural proteins that is essential for the replication of the virus. And in this after this viral polymerase is formed, then further the positive single strand RNA need to be converted into negative single strand RNA that can be uh, uh, then that negative single strand RNA can be converted or translated into the proteins, into the proteins for which it has been encoded with the help of cellular machinery. And along with that, the subgenomic RNA, that is the structural proteins that is necessary for the replication of SARS-CoV-2 virus, are also been uh, are also been translated parallelly. And this subgenomic RNA, we can call it as the four structural proteins, protein code, or we can say that four structural proteins RNA, that is nucleocapsid protein, spike protein, membrane protein, envelope protein. So at this place, the viral genome is also replicated, and this structural proteins are also replicated. After that, there is a segregation of pathway occurs in a way that the structural proteins, three structural proteins, that is spike protein, membrane protein, and envelope, will go to the endoplasmic reticulum for post-translation modification. And the post-translation modification that begins in endoplasmic reticulum is and glycosylation, and whereas the viral genome will and nucleocapsid will remain in the cytoplasm. Viral genome will bind to the cytoplasm and go directly to the ERIC, that is endoplasmic reticulum Golgi intermediate compartment. And from here, with the initiation of post-translation modification in envelope, membrane, and spike protein, it will also move to the ERIC compartment. And at this point of time, the nucleocapsid and the other structural proteins formed uh, combined together, and then further post-translation modification occurs from ERIC to Golgi pathway, and then they proceed towards the secretory pathway. Uh, and during this uh, process from ERIC to Golgi, the complete formation of SARS-CoV-2 virus occur, that can be transported, or exocytosis that can be that is capable enough for being transported to the extracellular matrix and can affect the other cell. And this process of transporting any proteins or any virus out of the cell is known as exocytosis, or transporting anything thing out of the cell is known as exocytosis. So this exocytosis occur, and then this SARS-CoV-2 virus can affect the other cells. Now let's focus upon the strategy, proposed therapeutic treatment for SARS-CoV-2. The basic strategy uh, for prevention of any virus is to avoid its interaction with the host cell, and best way to avoid its interaction is to disrupt either the SARS-CoV-2 uh, either the virus portion that interact with the host cell receptor, or the or modify the host cell receptor in a way that it will not create any negative effect on the host. At the same time, the efficiency of the virus binds to the host cell reduced. And this is the first strategy. First two strategy is also about the same. In the first strategy, what people are trying to do is to uh, cover this ACE2 receptor with the help of soluble ribosome receptor binding domain. And the second strategy is to use SCFV against ACE2. So both of these will when these are bind to the ACE2 receptor, the SARS-CoV-2 virus does not get any direct contact with the ACE2 receptor, thus not being able to uh, invade, not being able to invade the cell. And second strategy is the target the spike protein of the SARS-CoV-2 virus, so that it will reduce the replication. It will reduce the replication of SARS-CoV-2 virus within the host. And third strategy is uh, third strategy is a complicated strategy. However, it has been more popular nowadays, as it will prevent different section of the replication, to reduce the replication of SARS-CoV-2 within the host. This first strategy is to use and here is the target of the different drugs that are being used, and which strategy of the which portion of the SARS-CoV-2 uh, replication cycle are being affected. So here, first strategy is used to use antibody. First strategy is used to use antibody, that is SCFV against ACE2. What this helps? Uh, this this helps or prevents the SARS-CoV-2 virus from invading the cell. Though the budding light structure is formed, the SARS-CoV-2 virus will not be able to enter inside the cell because of the inefficient or improper interaction of ACE2 with the spike protein of SARS-CoV-2 virus. and and this is also this strategy is also enhanced by chloroquine, as chloroquine is seems to affect the glycosylation of the ACE2, that further involve in reducing the efficiency of ACE2 binding with SARS-CoV-2 virus. And second strategy is to use this aplemoid colchine vinorelibin uh, drug. What this drug does, this drug does not allow the release of viral genome inside the cell. So if the viral genome is not released, then the replication of the SARS-CoV-2 virus will not occur. And so this strategy has also been used, or also under investigation. And after that, the another strategy that has been used is to prevent the translation of viral genome. And many of us has heard about remdesivir, favipiravir, and ribavirin. So this remdesivir will uh, reduce the translation of the viral genome, whereas favipiravir will inhibit the translation of structural protein, and any inhibition of any one of these will impact its replication rate, will impact on its replication rate. And this ribavirin is also affect this recept ribavirin will also affect is also work in similar way this remdesivir and favipiravir works. That is, it also inhibit RNA polymerase that has been used during the translation process. And this bananin 5 hydroxychromoin, what it will does is it will prevent the initial translation of the viral genome to the viral polymerase. uh, and if this step is inhibited, then it will not it will it will not only prevent the replication of SARS-CoV-2, however it will also prevent the cell from being taken over by the SARS-CoV-2 uh, virus. And this some of the uh, sequence siRNA based therapy. This siRNA based therapy the will prevent the formation will prevent the proper folding or the structural proper structural folding of the structural proteins and the viral genome, uh, that will inhibit its efficiency to affect other cells. And the strategy what last strategy that has been presented in this paper is to prevent exocytosis with the help of chloroquine, as chloroquine not only prevents endocytosis as it will affect the glycosylation of the ACE2, however it will also prevent exocytosis, which is an essential step for the SARS-CoV-2 virus replication. So this is just an overview about what are the strategies that are being used uh, or under investigation to prevent the SARS-CoV-2 virus to affect human beings from affecting human beings. Okay, so that's all for this video. Thank you for your time.